Yes, you are too efficient. Okay, so good morning, everyone. Again, this is a very large pleasure for me to introduce this webinar. This webinar was organized as a bridge between two projects. The first project is AgriLoop, which was financed under Horizon Europe, Horizon Europe, and the second one is Cipromed, which was financed under the Prima funding scheme. The projects are similar in certain ways and different, of course. You will hear the two coordinators describe the projects. And then we will hear several speech about different aspects of the project, especially because both projects are dealing with plant residues or plant biomasses in order to extract proteins and other active compounds to produce food, feed, bioactive materials. And so what we are doing this morning is try to give some hints, some advances regarding the pretreatments, the way of extraction, the last news that are coming out from those projects. For all that need a certification of being present to this webinar, I'm here, just mail me. You have my email, either you Google it or you find it on the program. Email me and I can send you a certificate of participation. I mean, PhD researchers and so on. And also for the question, please write them in the chat so that we will answer them at the end of each speech, if any, or otherwise, if they come later on, we can forward them to the speakers and then they can answer you directly. So I think we can start. The first is Shaina Saou, I think.
Yes.
Yes, she is presenting herself and the project AgriLoop.
Thank you, Annalisa. Just share my screen. Do you see my screen?
Yes.
Yes.
Is it in presentation mode?
Not yet. Just click on that. Yes.
Now it's okay?
No, we don't see it in presentation mode. Yes, now yes.
Okay, so hello everybody. I am Shaina Zaouf. I am from INRAE at Montpellier, France. And I'm going to present briefly the project AgriLoop. So AgriLoop, it's an European-Chinese project. And so AgriLoop for circular reasoning for agricultural residue. And its main objective is to transform the agricultural residue into a portfolio of biomolecules and products. And this project is funded by the European Commission, by the Ministry of Science and Technology of China, and by the British and Swiss government. The coordinator on the European side is Nathalie Gontard, and in the Chinese side is Professor Aimin Shi. So for millennia, man has explored the plant and animal biomass available to produce food, energy, and materials, and so on. And with the discovery of the fuel or the oil energy, this activity has expanded, and so the many products have been produced, among them energy, plastics, fertilizers, and other chemicals. And this contributes, of course, to the well-being of the human being and their safety, but on the other hand, it disturbs completely the natural biogeochemical cycles, and the linear strategy of the production contributed to a large accumulation of waste. So this exploitation of fossil fuels and the accumulation of waste are the main causes of the global warming and the pollution and contamination, particularly with the micro and nanoparticles of plastics, of non-degraded plastic. And this makes it's more urgent to develop and implement projects to replace these fossil fuels with renewable resources for the production of the food and energy and the materials. And it's more important to exploit the unused residues, so agricultural residues, which represent 50% of the total weight of agricultural production, by converting them into valuable products capable of replacing the existing fuel-based product, or by creating new markets with new opportunities. So the main objective of the AgriLoop project is to produce a portfolio of products from agricultural residues, products that could be integrated again in the plant cycle through the biogeneration. So as Annalisa said, AgriLoop project is the Horizon Europe with an international dimension. It's gathering 35 partners, half academics and half private from Europe and China, with the common goal, increasing the value of the agricultural production of the two players. So the consortium come from all over Europe and China. We have 20 partners from 8 European countries. So you can see that it's from France, Italy, Spain, Belgium, Portugal, and so on. We have 2 partners from non-European countries, UK and Switzerland. And we have also 13 partners from China. And also here we have academic and private partners. So this is a kind of a project identifier. So the project is coordinated at INAE, Montpellier, France, by Nathalie Gontard, and in China, in Beijing, by Professor Aimin Shi. So the total budget of the project is over 12 million euro, funded by the European Commission, the Chinese Ministry, and the British and Swiss government. And the project started at the end of 2022 and it lasts 4 years. So here I present the overview of the project. You can see that from primary and secondary livestock or secondary residues. So we have selected some residues going from tomato, apple, grape, peanuts, and brewery, and so on. So we implement some bioprocesses in order to transform them. So the bioprocesses are the mild extractions, the microbial conversions, and we transform them into biomolecules, biochemicals, so molecules, for example, proteins for food and feed. And also we have bioconversions processes in order to produce the biomaterials. So in a strategy of zero waste, all the extraction and bioconversion residues are also used, and they go to anaerobic digestion in order to produce bioenergy, or are used as fillers in order to produce, to go to the productions of the bio-based material. So the objectives of the project, we have a main objective to strengthen the relationship between China and the European Union, to convert these agricultural residues, so from some selected feedstock, into valuable bioproducts. So the bioproducts including biomolecules and biomaterials, which will go to the agricultural sector also in order to replace some products from the fuel or fuel-based products, and also to apply a safe and sustainable by design approach for process and products, and to demonstrate innovative and sustainable value chain, and to achieve several environmental, social, and economic impacts. So based on a safe and sustainable by design, so we have to take into account before going to the bioprocess, to bioproduct, we have to take into account the needs of the stakeholders and the farmers and the consumers. And we have to ensure also the reduction of harmful contaminants. And taking into account all this, we can then develop the bioconversion processes and go to the bioproducts which benefit to the stakeholders and the farmers that we have consulted earlier before going to the bioconversion and the bioprocessing. And so to guarantee the societal adoption and long-term impact, AgriLoop aims to align the design of bioconversion pathways with the principle of circular economy. So before innovating conversion technologies, several parameters need to be taken into account, such as raw material flows, intermediate and product markets, chemical contaminants, environmental emissions, industrial input and output, and this will ensure a rapid acceptance of all the products that we generate. So AgriLoop relies on 4 scientific and technical work packages. The first work package consists in anticipating the complex circularities of the agricultural residues, and is based on the development of safe and sustainable by design to avoid or minimize the harmful impact on health and environment. The second work package is to improve the recovery of native molecules, so the extractions of these plant proteins, polyesters like cutin, suberin, and other bio compounds like polyphenol and carotenoid. And in the third work package, we are working on the bioconversion in order to produce microbial protein and microbial polyesters like PHA. And the final work package and to tailor final products to just needed requirements using a frugal design strategy, and also to accelerate stakeholder adoption of the processes and products developed as we be demonstrated by scaling up selected biorefineries. And all these work packages are linked together. For example, the work package 1 is the early guidance. It will benefit the other work packages, and the residues of work package 2 will go to work package 3 and vice versa. And work package 4 will benefit from all the processes and the products developed in the work package 2 and the work package 3. So this is an overview of the project. Thank you for your attention. And if you need to know more about the project, don't hesitate to connect to this media and to email Annalisa or Natalie or Aimin for further information. Thank you.
Thank you, Shaines. And so we may want, if you have any question, write them in the chat. If not, yeah, we can move on to Cipromed project. So I don't know if Christos can upload, share. Yes, perfect. Christos.
Okay, thank you, Annalisa. Can you see my first slide in presentation mode? Yes?
We see it not in presentation mode for the moment.
Let me try it again. Maybe now? Not yet?
Not yet.
Then I will stop share and try to share it again. it work now?
No, we see it not in presentation mode, but I don't
Okay.
know. Listen.
Should I insist trying, or should I start presenting it like that?
If you, well, I think we can see it like that.
Okay,
I
if you
don't know.
can see
As
it.
you wish, as you wish.
Because in my screen I can see it in full mode, in presentation mode, but I don't know why you don't see it.
So maybe it depends on what you are sharing, if you are sharing the screen or the PowerPoint.
Okay, then.
If you share the screen, then you should see the screen.
Okay. I will share the screen. And probably now.
Yes, perfect.
Okay. So, good morning, everybody. Thank you, Annalisa, and thank you also for all your efforts to organize this really nice webinars, and thanks for giving the opportunity to present Cipromed. I will be presenting Cipromed on behalf of the coordinator of this project, who is Professor Christos Athanasiou, who is professor in the University in Thessaly, here. As you can see from the title, Cipromed is about alternative proteins. So the title of the project is Circular and Inclusive Utilization of Alternative Proteins in the Mediterranean Value Chains. So it's a recently launched project. It's a PRIMA project, as you mentioned also in your introduction. PRIMA projects aim to reinforce partnership for research and innovation in the Mediterranean region. So the project was launched 1 year ago, 1st of May 2023. It has 17 partners from 10 countries. I thought it was a quite big consortium, but then I saw the consortium of AgriLoop with 35 partners, and I really realized what is a big consortium. Okay. So we have partners from all over around the Mediterranean basin, but also from Germany. And all these partners, you can see here their logos, 8 research organizations, including University of Bologna, Israel Institute of Technology, University of Torino, but also many companies. So it's a quite balanced consortium. Companies from the insect producing sector as Next Protein or Flying Spark, feed producers as LVs, microalgae producers as Alga Energy. So it's quite diverse consortium. So all these partners joined forces to address one question into one issue, which has to do with the dependency of the current agricultural production systems on protein imports. Most of the proteins that we are using in EU are imported. I'm talking about soybean meal or about fish meal, and these supply chains can be easily disrupted. We had several occasions recently with the coronavirus or with the crisis with the different wars. So there is an urgent need for EU to find viable, locally produced, if possible, alternative protein sources. And this is the big question that we try to answer within Cipromed. And there is just one goal, the main goal, to increase the stability of the Mediterranean agricultural systems. And to do that, we will be valorizing proteins from locally generated and agri-industrial side streams. We will be using brewer spent grain or different oilseed press cakes. And we will be valorizing these side streams, upcycling them and bioconverting them using insects and microalgae. And these are actually the 3 raw materials that we will be exploiting within Cipromed. We will be working with microalgae, both heterotrophic and autotrophic microalgae. We will be working with legumes that have been overlooked during the last years, like lupins or like faba beans. We'll be working with lupins and faba bean varieties that are intended to be used both for food and for feed. And of course, we will be working with insects, particularly two insect species, the yellow mealworm, Tenebrio molitore, and the black soldier fly, the BSF. And we will try to exploit these raw materials using novel techniques, and of course exploiting fermentation. There will be a presentation later on in this webinar about the importance of using fermentation to improve the attributes and the traits of the proteins extracted from these raw materials. I think this infographics illustrates what we will be doing in Cipromed. So these are the three raw materials that we will be using: insects, microalgae, and legumes. And in order to produce these raw materials, we will valorize, as you can see here in the upper left corner, different side streams, agri-industrial side streams, food processing residues, or industrial CO2 for the culture of microalgae. All these raw materials will be processed with novel processing technologies, novel extraction technologies, novel fermentation technologies, and even the residues of these extraction technologies will be exploited for the rearing of insects and microalgae. So we try to employ a zero waste approach, so nothing should be wasted. The same counts, for example, here, if you see here this loop for insect rearing, we will be rearing insects on side streams in order to produce insect protein to be further used in to formulate food and feed products. But even the byproduct of insect rearing, which is insect frass, this is actually the manure of insects, will be valorized as soil fertilizer for the production of legumes to close the loop. And at the end, the final output of the project will be different novel food and feed products. More specific objectives from the raw materials that I mentioned previously: insects, microalgae, and legumes, to extract high quality protein ingredients to be further used for food and feed applications. Using these protein ingredients, we will formulate new food and feed prototypes. Particularly with the feed prototypes, we will be conducting feeding trials with poultry, with fish, to see how these innovative novel feed diets will be affecting the performance of the livestock animals and the fish. And regarding the food products that we will formulate, we will also run human clinical trials to see the impact of these food prototypes on human health also. And of course, we will not forget to address also safety issues, regulatory compliance issues, as well as the sustainability of all these processes and products that will be produced. There will be a lot of LCA analysis, LCC analysis to determine the environmental and economical impact of the process and the products that will be generated. Consumer acceptance, consumer perception is also something really important. We will be running also different surveys, consumer surveys, during the 3 years of the project. And last but not least, at the end, we will develop specific business models and exploitation plans for the processes and the products that will be generated within the project. And just to give you an idea of what we've been doing, really rough and fast idea of what we have been doing during the last year within Cipromed, we have done a lot of trials with insects, insect feeding trials on valorizing different side streams. You can get just an indication here in this picture. These are different oilseed press cakes that we have been using to rear our insects, both the yellow mealworm and the black soldier fly. We've been conducting already several trials with frass. Frass, the byproduct of insect rearing, has been used as soil fertilizer for the production of both lupin and faba bean varieties, both in Greece. Here, for example, you see some pictures from trials with lupins in pot experiments in Greece, but also in the field. Here you can see some field trials in Morocco performed by partnered GDI, who is situated there in Morocco. And I will conclude my presentation with a really recent development within Cipromed. Really, I think it was a nice event. It was a workshop that was held yesterday within a project, within a conference that is dedicated to edible insect. This conference was held yesterday in Germany. It was called Insecta, and within this workshop, we ran a sensory tasting event with insect-based breads. You can see here on the right, our partner, Rainer Stolzenberger, baking insect-based breads, 2 different kinds of insect-based breads. And then there were more than 200 participants in the consortium, in the conference, that tried these breads and told us their perception. So we're looking forward to these results. The sensory trial was run just yesterday in the morning. And if you would like to learn more about the Cipromed project and follow our activities, we invite you to visit our website, Cipromed website, and also to follow us in LinkedIn by scanning these 2 QR codes. And of course, we are open for questions now during the webinar, but also afterwards. Please do not hesitate to contact us. Thank you.
Thank you. Thank you, Christos. If anybody has any question, please let us know. I'm monitoring the chat anyhow. Okay.
Maybe later.
Maybe later, yes. Maybe later. Okay, so then we can move to our next talk, which is Shyness again, which is now introducing some of the activities that INRAE is doing in AgriLoop project. So I let again Shyness. Yes, perfect.
Okay, so I will talk now about some activities that we are doing in our lab, and also we try to implement these activities in AgriLoop project. And it concerns a dry fractionation for enriched fraction in biomolecules. So dry fractionation is a dry process in order to enrich some fractions in biomolecules in order to reduce the use of solvents. So to define that the dry fractionation, it's a physical operation of dissociation and fragmentation, followed by a separation. So in the dissociation, the different constituents of the tissues are detached from the cellular matrix, and then the separation is based on the difference of the physical chemical properties of the different constituents. So according to this difference, the constituent could be separated by just a physical operation. So this process has the advantage to be a green technology. So we don't use much more harmful solvents and no effluents. And also it has the advantage to do not denaturate the structure of the molecules. So in our lab, so we have a platform which is called Planet, and in this platform we have several facilities for the grinding, milling, and the separation of the different constituents of the biomass. So we can work on food and non-food biomass. And as you can see here, you can see that we have several facilities for grinding, for example. So we have different mills, so knife mill, impact mill, ball mill, and all these mills can be used for different morphology of the biomass, and it could separate or fragmentate the biomass in different ways in order to facilitate their separation after that. And you can see here that we can have several impacts on the biomass particles. So we can have this impact, and we can have compression, we can have shear, and we can have also just a friction in order to scrap the surface of the particles and to remove only the superficial layer of the particles. And the ruptures are occurring when the strain is higher than the resistance, and the fractures are generated and propagated according to the fragility zones. So for the separation, we have several devices, but we can go from the very simple separation according to the sieving, for example. So we can use a sieve with different screens of the sieve, and it could be sufficient to separate different fraction with different morphologies and different compositions. And if it doesn't work, we have more sophisticated facilities or processes. And here I will talk about one process that we have and that we use is the electrostatic separation. So what is the electrostatic separation? It's based on the turbocharging of the particles. So according to the composition of the particles, so after grinding, we have these biomass particles, and according to their composition, according to the water content, they will be charged either by scraping on the walls of the tubes of the device, or scraping on each others, and they will be charged on positive or negative charge. And then according to the charge, they will attracted by an opposite electrode. So it's the opposite polarity electrode, and then they could be separated in this way. So I will give some examples that we use dry fractionation in order to enrich some fractions of biomass on biomolecules. So the first example that I give is the wood bark. So we have worked on Douglas pine. So the Douglas is a French resource. And in France, we have 400,000 hectares of Douglas pine, and the France is the first European producer. And the pine of this essence is rich on polyphenols, so on phenolic compounds, and it contains up to 10% of phenolic extracts. And we can find two types of polyphenols in this bark. We can have taxipholine, which is the flavonoid, so this is simple molecule, and we have more complex polyphenols, which are the condensed tannins. So we have these two types of polyphenols. So we have tried to separate these two polyphenols in the Douglas pine bark using the dry fractionation, because that I said before, it's more environmentally friendly than the conventional process. So you can see here, if you see the image of the bark, you can see that we have different tissues. So we have this inner tissue with this old phloem, and we have this outer layer is the young phloem. You have the young phloem, and embedded in between these two tissues, we have these white tissues, what we call the cork. So after just a manual separation of these three tissues, we have found that the taxipholine is located in the cork, and condensed tannins are mostly located in this young phloem. So we can find this. It doesn't work anymore. Okay, stops. Sorry, I will stop. Okay, now it's work. So we have found that, as I said, that the tannins are mostly located in this part, and taxipholine is mostly located in this white part, what we called effort. So we have applied the dry fractionation in order to separate the different tissues and to enrich the fractions on taxipholine and on tannins. So the first thing that we have done is just milling with a knife mill, and then a sieving in order to separate the different fractions. So we got a coarse fraction, which contained the most of the taxipholine. So 62% of taxipholine was located in this coarse fraction, and then a finest fraction was rich on tannins. So we have found 43% of tannins in this fraction. So we wanted after that to enrich more on taxipholine. So we just took the first fraction that we have milled, and we tried to apply a ball milling on this fraction. So, and you can see here that we have enriched more a fraction on taxipholine. So we got a fraction more rich on taxipholine, as 83% of taxipholine was located in this fraction. So by just a milling and a sieving, we could enrich a fraction with almost the total amount of taxipholine. Okay, stop. I'm sorry, it doesn't work. I think I will say like that because it doesn't work really. So and here you can see the chromatogram of this of the molecules. So we have here the taxipholine, and we have here the extension units of the tannins, and you can see that these fractions is really rich on taxipholine. So we have another example. It's now it's for food application. So we have also done the wheat bran dry fractionation, and this work has been done by the team of Xavier Rouault and Joël Abécassis. So you can see here that the wheat is the most consumed cereal in Europe, with more than 100 kilo per person per year. And the wheat grain is a complex structure composed of several parts. So you have the main part is the starchy endosperm. So that's we build transformed into flour. And then you have germ, and here you have all these bran. And the bran is constituted of outer layers. So we have aileron with 8%, and we have also the outer pericarp, and all these constitute the brain, which constitute 20% of the weight of the grain. So the purpose of the milling is to separate the brain of the flour. So the flour is used for the bread preparation, and we have also the bran. And the bran is now constituting more than 6 million tons produced each year in Europe. And it's mainly used for feeding, but it's under valued, mostly used for animal feeding, but it has a very interesting nutritional potential. So it could be used or exploited for other applications. So it contains the most vitamins, mineral, antioxidant, and antioxidant phytochemicals, and it can contribute to increase the nutritional quality of food. And you can see here in the microscopic analysis that you can have these pericarp with the insoluble dietary fibers and antioxidants. We have these testa and aileron with the dietary fibers and ferulic acid, for example, and you have also intercellular content, which are proteins, lipids, and antioxidants. So the main purpose to fractionate the wheat bran is to remove this outer layer. So the outer layer is composed with detrimental compounds. For example, the toxins, the heavy metals, pesticides can be present in this outer layer. And to remove also the phenolic compound would be responsible for the bitter taste, for example, for phytate and anti-nutritional also product, and to concentrate the interesting compound to produce ingredients enriched in aileron, known for its high nutritional potential, rich in dietary fibers and in antioxidants. So for that, the sieving was not sufficient, so we have to use the electrostatic separation that I have described before. So we have tested this electrostatic separation in a pilot scale after turbocharging. So the particles were separated in an electric field depending on their accurate charge. And the high percentage of composite particles in bran much lower purity of fraction. And we made 3 successive separation steps in order to have a good separation. So we can see here. So this is a microscopic images, and you can see that the negative charge was rich in pericarp material. So the layer that we wanted to get rid, because it's containing mycotoxins and pesticides, and all the positively charged fractions were rich in aileron, which the good fraction that we need for each richness in vitamins, polyphenols, and dietary fibers. So we can see that with these 2 examples of wood and wheat bran, that with dry fractionation by applying just simple processes, for example, such as sieving, or more sophisticated one, such as the electro separation, we can have rich fractions in the targeted molecules that we want to obtain. And for the extraction, we will certainly use a reduced amount of solvents. So this is the advantage of this process. Thank you for your attention, and if you have questions. And sorry for the presentation.
Okay, thank you, Shana. I have a question. Can I ask a question?
Yes, of course.
The question is, which are the limitations? So I mean, which type of feedstock is preferred? I see you have seeds and things that are dry, or do you have to dry them before anyhow subjecting? Okay.
Yes, drying is important, because if we have a big amount of water, the grinding is not performed very well. And also, if we have a fatty feedstock, for example, like seeds and also which contains oil, we can have some problems in the grinding. So sometimes we remove the fatty part before grinding in order to reduce the problems of aggregations in the mill.
Okay, so you need to be very much sure of which type of feedstock you are entering. Otherwise, you might have. Okay, that's very, very interesting. Thank you. Any other question? Yes.
Can I ask? Good morning. Shane, I follow a little what Annalisa was asking. I'm curious to know, so in the first stage that you are grinding, do you generate any heat? And I'm asking this to know if there is any chemical modification because of the heating during the grinding step.
Yes.
And the second question is in the fractionation, especially if you are considering the sieving, what is the lowest particle size that you can obtain?
Okay, so for your first question, we test the feedstock that we put in our mills. And sometimes, yes, we generate some heat. So if we see that the heat is, the temperature is too high, we can add some liquid nitrogen in order to just to reduce the temperature to do not denaturate the molecules and the structures of the molecules. And this is one method that we used. And for your second question, it was about
the smallest particle size.
Yeah, the smallest particles, we can go up to I think 0.001 millimeter in the sieves.
Okay.
Okay, thank you. Thank you very much. Any other question? No? Okay, if you
in the chat, there is a question.
No, this is me writing. Please write here. Okay. Or
okay, so
I don't see did you see question? No. Anyhow, if you have questions, write them, of course, in the chat, and then we can answer even later. Okay. Our next presenter is Jing Hin. He is part of our twin projects, AgriLoop China project. So I leave the floor to Jinghin so he can present himself and present his work. Thank you.
Can you see my screen?
Yes, yes, we see.
Okay.
Thank you.
Hello, everyone. My name is Xin from Institute of Science and Technology, Chinese Academy of Agricultural Sciences. The presentation I'm giving today is value-added utilization of the apple processing byproducts. Here are the contents. First is the background. Every day, people consume lots of food products. According to the literature, is that approximately we produce 23.7 million tons of food products every day. Along with the production of the food products, there are about 250 million tons of waste generated annually, with 5 billion tons of crop residues produced every year. So there are lots of food waste generated every day. The question is, how can we reduce the waste, whether we can reuse some of them? Currently, most of the food waste are used with low value added. For example, they can be used as fertilizer, feeds, biofuels. Only small portion of them are used with high value added in the terms of food nutrition and health, chemistry, and pharmaceuticals. So now the question is how we can increase the portion of the food waste with low value added convert to high value added. Taking apple as an example, according to the data in 2022, the annual yield of apple worldwide is about 1 billion tons per year. And China is almost the largest apple planting country. The data for China is about 50 million tons. And in China, most of the apples consumed as fresh fruit. Only small portion are processed, with liquid processed products as the main products. For instance, the apple juice, apple vinegar, apple mud. And with production of the liquid products, there are apple pumice generated. And only a small portion of them are used as feed, fertilizers, or fuels. Analyzing the composition of apple pumice, we can see that the pectin and cellulose content are very high, and also with certain nutritional compounds, for instance, the polyphenols. So the next part is the pectin and the polyphenols extraction and characterization. Pectin is the most abundant in the plant primary cell walls, and the middle lamella is a family of the heterogeneous polysaccharides. During the apple processing, there are little a small portion of the pectin will be released from the cell walls, and with further extraction, more pectin can be released and generated. The conventional extraction method includes Howtare extraction or with alkali acid assisted extraction. There are also other emerging extraction available in recent years. For instance, with chemical methods, the NACE method or the enzyme-assisted extraction, or with other physical fields assisted extraction, for instance, microwave, ultrasound, radio frequency, and etc., or combined extraction. Both the drying, pretreatment, and the extraction procedure may influence the structure and the functionality of pectin. Of course, also the extraction rate, cost, grain, and environmental impacts. So it's very important to optimize the drying, pretreatment, and also the extraction. There is an example done in our group in the previous years, that we use one pretreatment before the pectin extraction of extraction of pectin from peach pumice. We use the instant controlled drop treatment before the extraction procedure to increase the pectin extraction yields. And the results have shown that compared to the conventional extraction method, we can increase the pectin extractive yield of from peach pumice by more than 10%. So this is one example done from our group. And with extracted pectin, we can do the structural characterization, because all the extraction method and the different conditions, they all can have impact on the structure of the pectin. By doing, for instance, the monosaccharides composition analysis and MR analysis to check the structure of the pectin, the degree of esterification or molecular weight analysis, we can do the structural characterization. This is an example of the pectin, and you can see that they have different fractions, the so-called Hg, Rg1, Rg2, or Xg, and all different fractions have different functionality. They can have, for instance, antioxidation, anti-cancer, lower blood sugar, or antibacterial function. But they are all affected, can all be affected by the extraction, different extraction method or extraction conditions. So it's very important to study the extraction and the structure characterization in order to know the functionality of the pectin. Here is another example from literature. They have shown that different extraction methods do have impact on the on the properties of pectin from apple palmist. The results have shown that the extracted pectin yield was highest for microwave assisted extraction, and both the microwave assisted extraction and ultrasound assisted extraction pectin have higher GALA content and higher molecular weight. So this is one example from literature. And because all the extraction method and condition may have impact on the pectin yield as well as the functionality, it's also very important to know how it behaves during the metabolism. And from lots of study, we know that the pectin is very stable during the digestion in the oral space, stomach, or small intestine, which is due to the resistance of pectin to gastric acid and human digestive enzymes. So people also use these properties to apply in the controlled release of nutrients and the controlled release of medicine. But it's also it's also influenced by the pectin properties and also the pectin structure. So it's very important to know how it's affected by the extraction method and injection condition. Here is another example done by our group. This is a pectin extracted from apple. It's not from apple palmist, but this is extraction of pectin from apple. In this study, we have shown that the impact of extraction on pectin structure and in vitro gut fermentation. In this study, we have shown that the different extraction method, for instance, the hydrochloric acid extracted pectin, enzyme assisted extracted pectin, and water soluble pectin, they'll have higher molecular weight and a higher degree of esterification. And this study we also have shown that alkali extracted pectin have the highest short chain fatty acids, which is beneficial for human body. So all these results have shown that it's very important to not only to increase the extraction yield, but also to study the correlation between the structure and the functionality of pectin. Next is polyphenols. We have an analysis on the polyphenol content in different parts of apples, and our results have shown that the high polyphenol content were found in the peel and the seeds, which are parts of the apple palmist. So it is possible to extract the polyphenols from the apple palmist. The total polyphenol content in apple palmist is in the range of 2 to 6 grams per kilogram. The extraction method either can be conventional with the ethanol extraction, or similar to pectin, we also can do the physical field assisted extraction, for instance, ultrasound, microwave, high pressure assisted extraction, and so on. The chemical methods like biochemical enzymatic extraction, or the methods which we will apply in this project. To the extraction process, this is example of the extraction process of apple palmist polyphenols. The extraction to select the proper extraction and to study its effect on the extraction yield and the polyphenol monomers, and also the properties or structure of the polyphenols is also very important, similar to the pectin studies. So the the solvent type, solid liquid ratio, physical field, or other field assisted. It's all that should be optimized to optimize the extraction process of polyphenol from apple palmist. This is an example we got from literature, which has shown that the traditional solvent diffusion is time-consuming with low yields of polyphenol extraction. It also may happen the thermal degradation or oxidation because of the very long extraction time. The results have shown that with ultrasound-assisted extraction, we can have much higher polyphenol yield compared to other methods. This is a active polyphenol evaluation of the structure of different polyphenols and its functionality. The polyphenol metabolism also includes 4 parts. That's the release and breakdown of polyphenol from apples in the stomach and small intestine, absorption of phenolic compounds by small intestinal cells, transport in the bloodstream and redistribution in tissues, and then elimination via kidneys or re-excretion through bile and pancreatic juice. And finally, the microbial fermentation of unabsorbed phenolics. But also, as different polyphenols will have different behavior, which also largely affected by the extraction procedure. So, similar to pectin, it's very important to study the extraction the effect of extraction on structure and its association with the functionality of the polyphenols. The final part is the commercial application. Actually, nowadays there are already quite some application of pectin and polyphenols. It can be used for its nutritional function as a nutritional supplement, or due to its properties, it can, for instance, the pectin can be used as a food additive, which is already common use as a emulsifier or thickener, and for the protective low digestive of pectin in the oral stomach and the small intestine. It also can be used as drug delivery or nutrients delivery for its controlled release. Actually, there are already some commercial application, but the application from the effects from the waste, I think, is still limited. So in future, the work should be done to increase the portion of the extracted from the food waste to apply it in food industry. This should be the future commercial application. Okay, that's come to the end of my presentation. Thanks for your attention. Questions are welcome. Thank you.
Okay, thank you very much for this impressive work. Are there any questions? Can I ask a question, if you don't mind? Yes.
Of course.
Of course. So you are
welcome.
Sorry. Yeah. I'm asking a question in relation to your pectin extract, and is there any other polysaccharides that you can extract by using the same procedure?
Actually, now we are for this project we design a sequential extraction. We also plan to extract the cellulose, which I have shown in the composition analysis that the cellulose content actually is much higher than the others. So we are trying to extract them in sequence, but we don't have results to show yet. But in this project, we will that. Yeah.
Okay.
Yeah.
Okay, thank you. Any other question?
May I ask before my presentation?
Of course you can. Go ahead.
So it's very interesting, and thank you very much. I have one question. I think one of the major negative environmental impacts is not just the extraction, but also the precipitation with ethanol. So the alcoholic precipitation that you need to do to concentrate the pectin. Do you have any solution that can improve environmentally this step?
Yeah. Now we are trying to decrease the use of this solvent. So to try to use less solvent is one of the also one of the goal of this part of the work.
And it works?
Now currently I cannot tell the final result because it's still ongoing, but we are trying to use the physical fields assisted extraction, so try to use less solvent compared to the conventional extraction. Yeah, that's one of the goal of this this part of my work. Yeah.
Thank you.
Okay. Thank you very, very much for your presentation, Jingjing. So now we move to Avi. Avi is he will present himself, and he is part of the Cipromed project. So go ahead, Avi.
1 second. Do you see it well?
Yes, perfect.
Okay. Thank you very much. Thank you for inviting me. And indeed, I am a partner of the Cipromed project. And I come from the Technion, the Faculty of Biotechnology and Food Engineering in Haifa, in Israel. And I will talk to you a little bit about as Christos mentioned, we are strongly focusing on novel technological applications to improve our possibilities of using the alternative protein sources. And today I will talk about some of our results about the utilization of high pressure homogenization as a pre-treatment tool for alternative proteins. So pressure is quite a common parameter that is used significantly by the food industry. In retarding, there is a small pressure increase. And of course, in high pressure processing for pasteurization, there is a very significant pressure increase. And there is a region that can combine also elevated temperatures of pressures of up to 300 or even 400 megapascals, allowing a process that is termed high pressure homogenization. One of the reasons that novel solutions for alternative proteins is actually needed is that actually we have quite a significant range of potential plant or cyanobacteria protein sources, starting from cereals going via legumes that we will be working on within Cipromed, but also tubers and so on and so on. But many of those sources share similar challenges. And those challenges are stemming from the fact that the proteins in those varieties are not necessarily evolutionarily planned to have the same function as the proteins that are from animal sources that we want to replace. They do not have the same fibrous structures as the muscle in meat proteins, and also very commonly, they do not have the same aqueous solubility or emulsification properties that we can expect from dairy alternative or from dairy proteins. They are simply different. They are very often part of the energy forming complexes of the plant systems or storage proteins. Homogenization as a whole is a process that is commonly used by the food industry. The main concept is that we want is to disrupt droplets to form a better and more stable emulsion. That's what is commonly used for milk. But when we are talking about high pressure homogenization, then we can go to significantly higher pressure levels. If regular homogenization is 20 to 30 megapascals, usually resulting in droplets of very often of several microns and having a gap size of about 20 to 100 microns. UHPH works at pressure levels of up to 400 megapascals, and the gap size in the homogenizer is much smaller, between 2 and 30 microns. This results in very significant and intense mechanical forces, shear forces, turbulence, cavitation, and so on, significantly affecting different functionalities of the composition of the liquid that goes through the homogenizer. It also includes quite a significant temperature increase of about 14 to 20 degrees centigrade per 100 megapascals of pressure. Therefore, temperature also contributes to the observed effects on functionality. The technology was originally developed as part of an attempt to reduce the negative impact of heat for microbial inactivation. It was shown to be quite efficient in different systems. I will not go into this in depth, but keep that in mind. But we do know that high pressure homogenization can significantly support better and more stable emulsions with lower viscosities. But if you go to pressures that are too high, emulsion stability can also be negatively affected, and therefore pressure level should be tested and verified for the specific product in need. And when we're talking about the effect of high pressure homogenization specifically on protein structure and structural changes, not a lot is known. What we do know that protein aggregates, which are very common for plant proteins in aqueous solutions, are significantly affected by high pressure homogenization, and this effect starts already from quite low pressures. On the other hand, again, if the pressure is too high, we can also increase the aggregation due to the beginning of even up to the level of beginning of gelation. Mostly the quaternary structures are affected, although some effects also can occur on the secondary structure of the protein, resulting in improved emulsification. And today I will briefly show you two projects that we were working with high pressure homogenization for alternative protein sources. The first one is plant-based yogurt alternatives. So plant-based yogurt alternatives are often in a significant difference from the real dairy milk yogurt, simply because the proteins are quite not soluble, except, let's say, of soy protein. This results in the development of products that have quite a low protein content, or and the use of stabilizers that are responsible for the formation of the gellose network is required. In most cases, in alternative protein yogurts, the structure of the network does not stem from a protein-protein interaction as in dairy yogurt, but due to the presence of stabilizers or oils. One of the reasons is that if the particle of the protein separates a fish quite quickly during the time required for fermentation, we will not obtain a homogeneous yogurt alternative, which of course will not be a good outcome. Therefore, even if we can perform fermentation on the protein source, and the protein has an isoelectric point that is similar to the isoelectric points of dairy proteins, we still might not get a homogeneous yogurt due to the fact that during fermentation, we will have phase separation. And this is a thing that we wanted to solve by the utilization of high pressure homogenization. And we tested this by the application of potato protein isolate, which is quite soluble, but not fully soluble by itself. And what we have done is that we used 2 steps of high pressure homogenization. A first step was used to improve the protein functionality, and we had to keep the temperature quite low, otherwise we would get aggregation of the protein. And the second step is to form a stable emulsion with oil that will allow us to perform a standard lactic acid based fermentation to form a stable and homogeneous yogurt alternative. And we can see some results very briefly. If we did not allow the first step of homogenization just to break down protein aggregate, you could easily see that in the emulsion step, we have here quite a clear sediment, a very negative sensorial aspect for the consumer. On the other hand, if we used high pressure homogenization on the protein itself, we reduced the level of sediment, we increased protein solubility, and it was a beneficial effect in terms of homogeneity. You could also see that the high pressure homogenization slightly improved the whiteness of the protein emulsion compared to control. The emulsion was also much more stable against phase separation. You can see it by the creaming velocity that significantly decreased compared to the non-homogenized sample. And you can also visually see it at the end of the accelerated stability of the emulsion test, that at the 200 megapascals had the most stable high pressure homogenized emulsion against phase separation due to gravitational forces. Of course, this was correlated with the decrease in particle size of the emulsion. So later on, we fermented the product. We had a similar pH reduction as in the case for dairy, as in the case for dairy proteins, in the case for caseins. Lactic acid bacteria efficiently grew in the system. And again, the final product, in this case, not the emulsion, had a much higher stability due to the high pressure homogenization of the emulsion than the non-homogenized product. And you can see it both visually and as a function of the phase separation here after the, again, accelerated stability test. Sorry. Okay. When we looked at the obtained gels, again, we could easily see the sediment occurred that if we did not use an initial high pressure homogenization step, the increase in the whiteness and the quite homogeneous system that was at the end quite similar to the system you could get from a milk dairy. When we compared the textural profile, we also compared the rheological profile. We got very high level of hardness, so even better than milk proteins, and we could easily tune the level of hardness by the level of high pressure homogenization and the concentration of the potato protein isolate that we used. And as I've shown, there was practically no phase separation during the expected shelf life of the product. The final example that I will show today is the utilization of high pressure homogenization as a pre-treatment step for thermal gelation of spirulina proteins. So spirulina proteins is a very nice source of protein. It can have up to 70% of proteins per dry matter and additional beneficial ingredients. But spirulina protein gels are quite weak. And we thought that maybe by high pressure homogenization, we could improve the gelation of spirulina proteins. We first verified that indeed, even in the weak structure of spirulina proteins, indeed, the proteins are the responsible for the formation of the gel. We wanted to verify it because polysaccharides from algae may also have a significant contribution to gel formation, but indeed it is the protein in the case of spirulina protein extract. And by the application of high pressure homogenization, we could reduce significantly the particle size of the proteins, of the protein concentrate, increase the protein solubility, as you can see here, when we increase the pressure level from no homogenization to 50 megapascals. This also increased significantly the surface hydrophobicity of the proteins, thus allowing us to obtain significantly harder thermal gels upon heating to, I think, 75 degrees C. The effect, we believe that the effect is from the fact that by increasing the solubility and by exposing the more hydrophobic regions, we could obtain a more homogenized protein-protein network upon thermal gelation, allowing us to produce an improved texture of thermal spirulina gels. Important to note that further pressure increase to 100 megapascals did not improve neither the protein solubility nor the surface hydrophobicity or the obtained rheological profile of the spirulina protein concentrate during thermal gelation. We believe that this is due to the fact that if we further increase the pressure, we already start obtaining initial aggregation of the proteins due to the combination of increased shear forces and increased temperatures. An important final point is that we have a very specific high pressure homogenizer in our system, but it is well known that the heart of the system of high pressure homogenizers is the various designs of the valves for the systems. You can see here several systems that produce high pressure homogenizers, and they are quite different. And as a result, one of the most significant limitations or knowledge gaps that we currently have is that different valve design can result in very different outcomes on the structure and on the functionality of the proteins. We have also seen it in correlation with our previous very interesting talk when we did the same by using different valves of pectin. So the pectin changed differently. Therefore, more information is needed about the impact of the valve design on the high pressure homogenization process, and as a result on the functionality of the proteins obtained. At the end, we also lacking knowledge on both the environmental impact of this technology. There is a lot of energy wasted in this technology due to heat dissipation, and regarding the downtimes needed to optimal maintenance of the technology. So at the end, it is a promising technology that can assist in the successful utilization of alternative and renewable novel protein sources. It can decrease aggregation, increase solubility, increase surface hydrophobicity. It allows us to produce better fermented plant protein products, but pressure levels and processing parameters, including also the design of the valve itself, must be better understood to optimize the engineering concept of these technologies. And I thank you for listening and open for questions if you may have.
Thank you, Avi. Thank you very much. Is anybody having a question?
I have, hi, Javi. Thanks for the presentation. I have two questions. One is very technical because you said that in the example that you have given with the plant-based yogurt, that you had oil.
Yes.
To get a more stable emulsion before the lactic acid fermentation. What is the percentage of oil that you are introducing, and how does that affect the end quality of the yogurt?
Okay, so I haven't shown you those results due to the lack of time, but we have tested and published already with oil ranged from 1 to 10%.
Okay.
The results that I've shown you today are 3%, which are common in yogurts. It works for all. It works well for all oil levels tested, including the 10%. At the 10%, we actually got the most significant impact of high pressure homogenization, again, due to the better emulsion, and also the TPA was the highest hardness with higher oil level. We did not have still a sensory test by human panelists. We do not have, our homogenizer is not food grade.
Okay. And the second question is about, so you show that there is very different types of homogenizers. And I think in your slide you have even systems that are based on microfluidics.
Yes.
And my question is, what is the downsizing in those systems? So what is the minimal amount of sample that you can use in those microfluidic systems, if you have idea?
Well, we have a microfluidizer here in my university, not in my lab. You can work with tens of milliliters. So
Okay.
let's say 100 milliliter will be good enough. My unit has a volume of 9 milliliters per run. But
Okay.
usually the problem is the upscaling, not the downscaling in those systems.
Yeah, I know. But to answer some of the challenge that you presented, the downscaling is also very interesting.
Yeah. But usually it's in the levels of tens of milliliters, I think, should work.
Okay. Thank you. Sorry, I was fighting with my computer because it was dying the battery. So sorry. Thank you. Thank you so much.
Okay, thank you. Any other question? I have maybe not so intelligent question, but anyhow, I make it. The question is, I presume that you have to start with very pure proteins in your treatment. So the question is, how much pure do these proteins have to be? Can you use, no, sometimes when you extract proteins, they are not 90% of your fraction.
Okay.
So is there any other contaminants, possible contaminants that you, because I saw your cyanobacteria extracts, they were blue, so I presume you have extra something in there also. So what I think, what my question is, how good they should be these proteins, so how pure, how homogeneous should be to be very efficient in your system, or any specific contaminants that you don't want to see for any means?
So for the yogurt alternatives, the concept was a bottom-up approach. So I wanted to make the product similar to the dairy alternative, so to the dairy. And dairies have proteins, sugars, and oil as main ingredients. And that's why I started with a pure protein. On the other hand, you can use any dispersion in the system. So I have worked with very diverse and very unpure dispersions, even up to the level of particles. If I use particles, I might be in a problem in terms of maintenance of the system. So again, scale up, but it works. But as long as my system is soluble or well dispersed, anything goes. You don't need to have a pure system.
Okay, but this will affect, of course, the last properties that you are looking for. Okay.
Of course.
Okay. It's a question of having a mediation of what you have and what you want. Okay, thank you so much.
Thank you.
Annalisa, can I make a last question?
Of course you can. Go ahead.
Thank you. You mentioned in the beginning that this technology also had some interest on killing potential contaminated bacteria.
Yes.
But you also show in the slide that the range of temperatures that you obtain are around 23, the maximum temperature. So my question is how, at least if I read well in the slides, so my question is, how can you get some sterilization effects? Is because the bacteria burst with the pressure, so it's not related with the temperature for sure.
So the number that you have seen, the 23, is the temperature increase per 100 megapascals. So, the temperature increase could be quite significant. We do control it by controlling the inlet temperature. So I had to reduce the temperature below room temperature to allow optimal functionality for my protein. But I can also start with 50 degrees C, for example, and then when I apply the high pressure homogenization, I could go even above 100 degrees C, allowing sterilization. The microbial decrease in the systems is known to be a combination of both thermal effect and the shear effect, and the shear and the cavitation effect. It's a mix of all effects, and it works. I've seen it in milk, and many others have shown it in juices and many other systems.
Okay, thank you.
Okay, thank you. I think we can have 10 minutes break, and we will, well, 8 minutes at the moment. We will resume at 10:50 Italy time, so it means, I don't know, you do it, your conversion. 10 minutes, okay? Cristina, se vuoi provare, prova.
Sì, grazie, mi senti?
Ti sento? Vai.
Ok,
Ti
perfetto.
sento bene e puoi provare a condividere lo schermo, lì dovresti avere un quadratino verde.
Sì, provo a condividere, vediamo direttamente la
Prova.
presentazione.
Se metti in presentazione.
A posto?
No, sì, è perfetta, vai indietro di una che vediamo il titolo. Sì, sì, funziona, io la vedo perfetta.
Ok, perfetto.
Ok, dai, a
Grazie
dopo.
mille.
Certo, figurati. Okay, so I think we could move on, if you don't mind. May, I saw you before. Are you ready? Yes. So if you can share.
Okay, can you hear me?
Yes, I can hear you perfectly. So if you
Okay.
can share, okay. So May is a part of our AgriLoop China project. Okay, here you are. Yes,
Yeah.
you put it in presentation mode.
Wait a moment.
Oh,
Sorry.
yeah. No problem.
So can you see my screen?
Yes, yes.
Yeah,
We're
okay.
waiting for the presentation mode.
Okay.
Okay.
Thank you, Professor Tosoni. It's my honor to share the high-value food components from potato starch processing waste in here. Before my presentation, I will introduce our team with a few words. Our team is the root and tuber crops food science and technology innovation team in Chinese Academy of Agricultural Sciences. There are 40 members in our team, including 3 professors, 2 associate professors, 1 assistant professors, 2 postdoctoral researchers, and more than 30 master and PhD students. The main research task in our innovation team is comprehensive utilization technologies of root and tuber crops processing byproducts, processing technologies of nutritional root and tuber crops products, and post-harvest preservation technologies of root and tuber crops. In order to ensure food safety, improve human nutrition and health, and realize full utilization of root and tuber crops. Today, I will introduce some research achievements of comprehensive utilization of potato starch processing byproducts. First is the background. As we know.
Excuse me.
Yeah.
Excuse me, Mama May, your screen doesn't move. It's still in this first page.
Now it is moved or not?
Nope.
No, you need to share the screen, not the PowerPoint. When you share, you select the screen, desktop, and not the presentation. So in this way, it moves. So yeah, perfect. Now it's moved.
It's moved?
Yes, we see planting area and production of potato. Okay?
No, but I cannot move my PowerPoint now. Wait a moment, I will stop it and again.
Yes, share the screen, not the PowerPoint.
Okay.
Try to move it.
But I cannot move my PowerPoint.
We see your mouse moving. This is what we see.
Oh, okay.
Yes, now it's moving. Okay, perfect.
I'm sorry, so sorry.
No worries. Go ahead.
Yeah, okay, I will start. First, I will introduce the background. As we know, potato is widely cultivated in the world, mainly in Asia, Europe, and America. For the China, the planting area our potato is about 5.7 million hectare, and the production yield is about 95 million tons. For the Europe, the planting area is about 4 million hectare, and the yield is about 98 million tons. So potato is can be defined as a bridge crop along the Belt and Road. Potato is rich in nutritional composition, for example, starch, dietary fiber, vitamin, and minerals. It also exhibits physiological activities such as lowering blood pressure, anti-cancer, and antioxidant. So it is recognized nutritional and healthy food by many countries. But in China, potato is mainly used to process starch. About 4.5 to 5.5 tons of wet residues and 10 to 20 tons wastewater are generated from 1 ton of starch production. Until now, about 80% residues and wastewater are disposed, resulting in serious environmental pollution. So whether the residues and wastewater can be used as high value components to added their value. Our previous studies showed that potato residues are rich in starch, total dietary fiber, and insoluble dietary fiber. And the content of cellulose and pectins in potato residues is about 21% and 10%. For the wastewater, it was reported that potato starch processing wastewater contained 2% to 5% dry matter, of which 1/3 is protein. And the essential amino acid in potato protein is accordance with the standard of FAO WHO. So potato protein has greater potential as a food product ingredient because of the high nutritional value. So our aim is to optimize the extraction technologies of pectin and cellulose from potato residues and the protein from wastewater, then investigated the structural properties, physical chemical properties, and physiological activities in order to establish production line for ecological cycle. The research progress was as follows. First is potato pectin. The effects of 5 kinds of acid, such as hydrochloric acid, sulfuric acid, nitric acid, citric acid, and acetic acid on the yield, emulsifying properties, and physical chemical properties of potato pectin was investigated. We found that the yield of potato pectin with citric acid was the highest, followed by the hydrochloric acid, while the potato pectin obtained by the hydrochloric acid showed the highest emulsifying activity and stability. Further, the protein content of potato pectin extracted with hydrochloric acid is comparatively lower. So the hydrochloric acid was chosen as the better acid reagent for potato pectin extraction. Then response surface analysis was used to optimize the extraction conditions of ultrasonic and microwave-assisted acid method. The optimized parameters were as follows. Ultrasound power was 50 watt, temperature was 93 degrees centigrade, pH was 2 and the time was 50 minutes. The yield of potato pectin was more than 22% under pilot scale, which was much higher than that of pectin obtained from conventional hydrochloric extraction. That was 90.72%. The microstructure of potato pectin was short chain, and the potato pectin was rich in galactose and galacturonic acid. In addition, the degree of acetylation of potato pectin was more than 50%. More than 17%, indicating maybe their potato pectin showed higher emulsifying activity. Then we also compared the emulsifying activity and the stability of potato pectin under different potato concentration, oil phase volume, and pH. The results showed that higher emulsifying activity and emulsifying stability was observed when potato pectin concentration was 2.5% to 3%, oil phase was 50%, and pH was 5. We also investigated the physiological properties of potato pectin. The results showed that potato pectin and high hydrostatic pressure assisted pectinase modified potato pectin showed higher lead adsorption capacity, which was higher than the commercial citrus pectin and some other carbo-rich adsorbents. The possible mechanism was related to the chemical adsorption, physical adsorption, and cation exchange. The next is higher fiber potato powder. First, we investigated the nutritional composition of different potato residues from 10 potato varieties, and found that LT5, Helan number 7, Zhongshu number 5, showed the highest nutritional composition. The waste potato residues was dried using microwave vacuum drying and superfine grading, then mixed with whole egg protein and cornstarch to produce the high fiber potato powder. This powder contained lower total sugars and higher total dietary fiber compared with the commercial potato flakes. We also investigated the application of high fiber potato powder. We found that after the addition of 30% high fiber potato powder to steamed bread, the specific volume color is comparable to that of wheat steamed bread. So the high fiber potato powder can be used as staple and snack foods. Now the high fiber potato powder has achieved industrialized production in China. The next is potato cellulose nanofibers. First, we also investigated the extraction parameters for the CNF. The results showed that the ultrasonic time was 15 minutes. The power was 125 watt. The homogenization cycles was 4 times, and the pressure was 40 MPa. The yield and Z potential of CNF was the highest. We also compared the microstructure of potato CNF with commercial CNF prepared by mechanical method and tempo-mediated oxidation method. The results showed that the diameter distribution of 3 kinds of CNF was the nanoscale range. The diameter distribution range of potato CNF, mechanical CNF, and tempo CNF are 20 to 60 nanometer, 20 to 130 nanometer, and 10 to 5 nanometer respectively, with average diameters of 36.41 nanometer, 56.26 nanometer, and 25.77 nanometer. It can be seen that the diameter distribution of potato CNF is more uniform, and the length of CNF is in the micrometer range, while tempo CNF have a shorter length. Then we also investigated the microstructure and thermogrammetric analysis of potato CNF. The results showed that CNF prepared with high-pressure homogenization process still maintain the basic structure of type 1 cellulose. Some crystalline regions are destroyed, resulting in a decrease in crystallinity from 53.01% to 35.44%, and the maximum thermal degradation temperature of potato CNF is comparable to that of mechanical CNF and superior to tempo CNF. The CNF showed better rheological properties, so it can be used as a fat substituent. The mixture of potato CNF and potato mesh can replace 40% of oil, which can be used in the processing of fat-reduced mayonnaise. Our results showed that the color, taste, and stability of the obtained fat-reduced mayonnaise was comparable to that of commercial mayonnaise. Now, the CNF was also achieved industrialized production in China, and the yield is about 20 grams per 100 gram potato residues. The next is potato protein. The potato protein can be extracted from potato starch with water. The purity of potato protein was more than 60%. The structure of potato pectin was also investigated. The peak one is protease inhibitors. Their molecular weight included 21, 22, and 16 kDa. And their peak 2 is pectin, with their molecular weight was about 43 kDa. We also investigated the physiological properties of potato protein. We found that the normal mice food pads were light yellow, while the food pads of wheat gluten protein sensitization group was dark blue, and the potato protein sensitization group showed light blue, indicating that the vascular linkage caused by potato protein was significantly lower than that caused by wheat gluten protein. And the histopathological analysis of intestinal tissues and lung tissues also showed that the pathological changes induced by potato protein was also not severe. We also found that the potato protein showed higher antioxidant activity, and the high pressure and the high hydrostatic pressure modification increased the antioxidant activity. the cell experiments showed that the potato protein can be inhibited the proliferation of colon cancer. And high hydrostatic pressure also increased the inhibitory effect of potato protein and the proliferation of colon cancer. So now we introduce the application aspects of the above high value food components in potato waste and residues. It can be used in low glycemic index and low fat foods, and it can be also used in degradable food package. For the health products, it can be used as chewable tablets or solid beverages to enhance immune and gut microbial and antioxidant activity. For the cosmetics, it can be used as cream, facial mask, and lipsticks in order to achieve anti-aging, antioxidant, and moisturizing. For the medicines, it can be used as soft capsules and injections in order to achieve the regulating glycemic, blood lipids, and pressure. In the future, we also need to further develop green intelligent extraction technologies for high components and expanded their application scope in foods, medicines, and cosmetics. That's all. Thank you.
Okay, thank you. Thank you very much for your nice work. Are there any questions? If not, I have one. Can I ask, related to your proteins from potato?
Yeah.
Maybe I missed something, but maybe not. I don't know. Did you see if you have the protein, the whole proteins, or you have peptides, oligopeptides in your extracts from the wastewater? So are they integral protein or pieces of proteins?
Pardon?
Did you see, did you measure the amount of peptides you have in your extract? So are they all integral protein or peptides?
Peptides from potato protein, right?
Yes.
Actually, in our team, we also prepared the peptide from potato protein by proteins enzymatic hydrolysis.
Yes.
Yeah.
So what I see, normally we see that the antioxidant activity increases when you have peptides. So that's why I was asking if you find high antioxidant activity. I was asking if you also have peptides in your extract. But yeah, okay.
Yeah.
That might. Thanks. Any other question? Okay. Of course, you always, you know that you can post questions. If you have them, we can forward to Megmay. And thank you so much. Megmay, can you upload your presentation in the folder that I've sent you the link, or either you send it to me and I upload it?
Okay,
So,
of course.
Okay, thanks. Thanks so much.
Thank you.
Thanks. Okay, so the next one is Arthur, which I already see him. And so please go ahead, Arthur. Thanks.
You are seeing my...
That's perfect. Go ahead.
So, good morning. My name is Artur Bento. I'm a researcher from the Cristina Silva Pereira lab. And today I will talk about how we can valorize the tomato pomace obtained from agro industries for development of cutaneous materials. So in the last decades, large-scale production of plastics increased greatly, especially as durable petroleum-based materials have been replaced by single-use materials. Every year, millions of tons of plastics goes to the ocean, creating a huge impact. This reality is daily exposed on the media, from floating islands in oceans to microplastics in the gut of fish. It is an urgent need to develop alternative sustainable process to ensure a greener and bio-based future. Most the plastics are not biodegradable. This includes petroleum-based plastics such as PET or polyethylene, and one alternative to replace the plastics can be explored the plant polyesters. I will start by showing a proof of concept for the production of antimicrobial oligomers from cutin-rich materials isolated in a single step from tomato pomace to generated by agro industries. In Europe, was processed about 10 million tons of tomato fruit. That amount generate about 0.45 million tons of pomace residue. Portugal is the third largest processor in Europe, where the derived pomace residue is mainly used for animal feeding. And several agro-food industries generate high amounts of readily available sources of biopolyesters through their residues. Accordingly, their usage contributes to accomplish the sustainable development goals set by United Nations by supporting a circular economy which aims to create novel chains of products from residues, reducing overall residues produced by industry. So the cutin is a plant polyester present in all land plants as a major component of their cuticle and covering all the aerial parts and fruits of all land plants. The cutin is deposited over the polysaccharide cell wall and is covered by boxes. Our group have been exploiting a way of extracting these plant polyesters that avoids major cleavage of ester bonds. By doing this, we can extract these materials as a polymer. In our process, we use the choline white, a biodegradable and biocompatible ionic liquid, to isolate the plant polyester. In the first step, we mix the plant sources. In this case, it is the tomato pomace, a residue provided by agro industry. We mixed with the ionic liquid at 100 degrees Celsius during 2 hours. After this process, we have a filtration step. Then we can isolate the cutin as an insoluble part, and we obtain a filtrate that have the polysaccharides and waxes present in the cuticle. In the AgriLoop project, we changed this old process by replace the DMSO by grass solvent. In this case, we use the ethanol, and the ethanol can be recovered by evaporation at low pressure. And the next step is to provide this filtrate to another partners of the AgriLoop project to take advantage of some to study some proteins or polyphenols that can be solubilized in the ionic liquids. This schematic representation highlights the main chemical groups present in cutin. We have the cutin is constituted by primary and secondary aliphatic esters bonds. Acids have some hydroxyl groups and epoxide. The ionic liquid extraction preserves the primary and secondary aliphatic esters. and the acylglycerol ester bonds are partially cleaved. However, this type of bond is present in a lower amount in the cutin. Again, this ensures that we can recover a large macromolecules polymeric structures that are insoluble in most organic solvents at room temperature. We produce the cutin oligomer mixtures, the COMs, by depolymerization of cutin polymer obtained by the ionic liquid method. Herein, the cutin polymer was hydrolyzed by two different methods to generate these COMs. The first one is using alkaline hydrolysis, and the second one is using methanolysis. After the hydrolysis, we filtrate to the cutins to remove the cutin's remains. We acidify and we have a centrifuge step to separate the products into different fractions. One that is soluble and another one that is precipitated after centrifuge. The impact of the COMs on bacterial viability was studied on two model strains. The one is a gram-negative, the E. coli, and the second one is Staphylococcus aureus, that is a gram-positive bacterium. We measure the cellular viability using the number of colony-forming units, the CFUs, and we measure the medium turbidity, the OD 600, also relative to the control. These two techniques offers information about the bacterial integrity throughout the exposure on the COMs. The COMs impact on bacterial viability was studied in the three different concentrations. Overall, the results revealed the significant differences in concentration-dependent bactericidal effects against the two species. Some data points show significant variability between replicates, which is most likely related to the heterogeneity of the COMs. The bactericidal action of COMs against SCRs increased proportionally with the concentration, suppressing the bacterial growth by about 84% or 97% at the highest concentration that we tested. In the case of E. coli, the viability of the COMs reduced to some extent the viability. However, the activity did not show a linear dependence concentration and we achieved the maximum viability reduction at the lowest concentration, and we have a reduction of 45%. We analyzed the chemical features present in the COMs by the nuclear magnetic resonance. Here are depicted the proton-carbon HSKC of the COMs. We can observe the presence of signals assigned to the presence of the primary and the secondary aliphatic esters. These two signals are present in all the COMs. So the COMs contains the oligomers linked through these two types of ester bonds. And besides the presence of these signals, we can observe the presence of the hydroxyl groups and some alkene groups in our mixtures. Each COM was quantitatively analyzed using NMR, especially using phosphorus NMR to quantify the free acids and the free hydroxyl groups present in these mixtures, and using the proton NMR using internal standard to measure the quantities of each ester type bond present in all the COMs. And the amount of primary and secondary aliphatic acids can influence the antibacterial action. The COM1S are highly abundant in the primary esters, and the COM2P are enriched in the secondary esters. And the COM2S have an equal quantity of each ester type. We also analyzed the COMs by GCMS, and we studied the ratio of each free monomer quantity before and after total hydrolysis of all the COMs to identify the building blocks that constitute the oligomeric structures. we detected that all the COMs had free monomers, accounting more or less between the 25% to 40% of their total mass. And alkanoic acids are the most abundant free monomers in these mixtures. The minimal inhibitory concentrations against S. aureus and E. coli reported in the literature were compared to the maximum concentrations that we can find in the different COMs. The best bioactive oligomers against E. coli are these three. Both the COM1S and the COM2P are essential composed mostly by the 1016 dihydroxide hexadecanoic acid, known as the most abundant building block of the cutin. However, the COM2P has twice much secondary esters than the primary aliphatic esters, and the COM1S has a lot of primary aliphatic acid in comparison with the secondary aliphatic esters. And the non-adioic acid is a possible building block in the COM2S, in the active oligomers, which includes similar proportions of both acid types. Now I'll change the subject of my presentation. I will talk about the work developed in the AgriLoop project, more essentially in the work package 4. And we began by applying the concept of getting these mixtures, oligomeric mixtures, to a different tomato pomace. In this case, it was provided by TomaPaint. We studied the antimicrobial activity against Staphylococcus aureus, and we found that the cutin pills produced by our method using ionic liquids had a little effect in the bacterial growth. However, the COMs had a high activity like we observed in our. So, and one of the goals is to functionalize polymers with these mixtures, oligomeric mixtures from cutin, to add antibacterial action and create functional materials. And we received the PHBV from INRE in France. And the strategy is to solubilize this polymer with COMs in the same solvent and then evaporated them to generate the films. We explored multiple solvents to solubilize the COMs, including the best for solubilizing this polymer. However, these mixtures are only solubilized in ethanol, acetone, and methanol. And the image shows a trial that we to generate a material including this polymer and the COMs, and shows the poor dispersion of the COMs across the surface of this polymer. The second polymer that we investigate to produce materials was the carboxymethyl cellulose, which we dissolve in water ethanol mixtures together with the COMs. The film was prepared by solvent evaporation. However, the major issue is the solubility of the film in water, which makes it impossible to access the antibacterial activity of these materials. And the following trial, it will involve heating of the materials to certify some of the COMs with the cellulose to reduce the film's solubility in water. So to conclude, we show a proof of concept of a tomato pomace to processing strategy for the production of bactericidal mixtures active against bacteria by using a mild depolymerization of cutin polymer that will release diverse oligomers monomers that display broad bactericidal activity. The COMs are active against S. aureus, and we show a concentration-dependent effect. And not all the COMs are able to kill E. coli. Activity is correlated with the presence of oligomers. Importantly, for the large majority of the COMs tested herein, the observed activities against either model bacterium cannot be simply explained by the presence of the free bioactive monomers, and the composites using the polysaccharide-based polymer, and comps are under development. So I want to acknowledge the funding and the collaborators and for your attention. Thank you.
Okay, thank you. Thank you very much. Any questions by the audience? No. Let's see. Can I ask you instead a question? I saw that thinking about what TomaPaint is doing, so they basically use the coating to coat food containers, you know, the tin of the tomato sauce or so on. Do you think your extraction, of course, does have give rise to cutin that has a lot of biological activities that I presume cutin from TomaPaint doesn't have? So do you see in the future applications other than coating or packaging or something like different than the simple packaging or the food application in the packaging?
Yeah, I think if you can take the advantage of the properties antimicrobial of the cutin, and if we know how to change some properties of the cutin, like make a selective depolymerization of the different ester bonds, we can have a very good material, resistant, and maybe we can have the antimicrobial activity in the same material.
Yeah, but you can say you can do packaging, but you could also do, I don't know,
Yeah,
pharmaceutical components.
yeah, I think we can. I think we in the future make different things and different packaging, or I think it's. I think the cutin.
So beyond AgriLoop, by the way. Okay. So that's a lot of work. Yeah, that's very nice, very nice. Thank you. Any other question?
Yes, I have a question.
Go ahead.
Why do you need to depolymerize cutin to have this antimicrobial activity? Is the polymer cutin antimicrobial as well or not?
I think the cutin can have some antimicrobial activity, but not against these two bacteria. I think the mechanism of to kill this bacteria can needs that some acids, free acids on the surface to have that capacity to kill these bacteria. But if the cutin is in the near inactive, it is not able to kill this bacteria, but in the plant is used to defend against another organism. So.
Okay, thank you.
So this gives rise to another question from my side. So if you change the bacteria, I think we presume we use another gram-positive or gram-negative bacteria. Do you think you need to find out another mechanism, or it is comparable because of the outer layer of the bacteria is similar composition?
The bacteria, I think the mechanism can be the same. But if you against fungus or another that normally threats the tomato plant, I think it can not. It's not needed to break down.
Okay, thanks. I think we can move on to next talk, which is from Maura Ferri. University of Bologna.
Yes, I'm here. I try to share my screen. Yes. No, this one. Can you see my screen? Okay. Hello, everybody. I'm Maura Ferri from the University of Bologna. I will talk you about plant proteins for sustainable future. This topic is connected with several sustainable development goals, at least with the goals number 2, zero hunger, number 3, good health and well-being, and number 12, responsible consumption and production. Before start with the presentation, we have to clarify 3 points, three key points. The first is the increasing world population and the future protein demands. The second is that food production has a large environmental and climate impact. And number 3, billion tons of proteins are wasted every year all around the world in food production chains. So let's start with the future protein demands. As you already know, world population is increasing, and it is expected to be about 10 billion in 20 or 30 years from now. And it would be impossible for such a lot of people to consume the amount and the type of proteins typical of current diets in North America and Europe. Accessible, affordable, healthy and sustainable proteins is critical to human nutrition and world economic development. Moreover, the food production has environmental and climate impact. As you can see in the graph, each type of food generates greenhouse gas emission, but the plant-based foods are in the lower part of this graph. It means that plant food has a less environmental impact. In this picture, you can see 2 inverse triangles. The first is the Mediterranean diet, and the second is the food impact. As you can see, the food that are suggested to be eaten frequently are the same food that has a low impact, and vice versa. European Union has initiated a protein strategy focusing on protein-rich crop cultivation, development of alternative proteins, and shifting the import-export balance of this product. This transition implies not only substituting one protein source for another, but represents a paradigm shift toward more sustainable, socially acceptable, and nutritionally equivalent or better protein alternatives. We also know that currently, approximately 1.3 billion tons of food are wasted per year all around the world, coming from all the different steps of the production pipeline, as you can see in the graph, from the primary production to the consumption. But these food production residues can be considered source of alternative proteins. In the picture, you can see as example 3 different agro-industrial residues and their chemical composition. You can see tomato seeds that we will meet again in this presentation, wheat bran and olive leaves as examples. For example, in tomato seeds, you can see that the major component of these residues are proteins. So they can be valorized. Moreover, alternative proteins can have health effect in human diet. For example, the alternative proteins from plant origin can reduce the mortality in humans up to 5%. That is a very good result. Moreover, proteins can be cut in fragments called peptides. Food-derived bioactive peptides are specific protein fragments that are inactive within the sequence and the structure of the parent protein. These fragments can be released during the human digestion by the human enzymes or the microbiota. They can be released in food processing, for example, in case of fermented food, and these peptides may exert various physiological functions beneficial for human health. These peptides have been found in almost all dietary proteins, and generally the most active ones share molecular weight lower than 10 kilodalton. Please remember this number, 10 kilodalton. And they are composed by 2 to 20 amino acids. Here you can see some of the potential bioactivity of these peptides. They can have effect on cardiovascular, nervous, gastrointestinal, and immune systems in humans. Now I will give you 4 examples coming from 4 different European projects in which we worked in our lab about plant proteins for a sustainable future. Of course, the first project is AgriLoop. You already know AgriLoop, so I will skip the presentation. In the frame of AgriLoop, we are working on with 4 different feedstock. 3 comes from tomato sauce production, and they are whole tomato residues, tomato peels, and tomato seeds. And the first comes from the beer production, and it's called brewery spent grain. We digested all these feedstock with the commercial enzymes. We tested the different proteases and or plant cell wall hydrolytic enzymes. Best performing enzymes were identified and processes were optimized. Here you can see an example of gel electrophoresis analysis that confirmed peptide release in presence of the proteases. In particular, as you can see in the circle, low molecular weight peptides are present, and if you check with the molecular weight marker over there, you can see that these peptides have a molecular weight between 8 and 12 kilodalton. As I told you before, 10 kilodalton is considered the best range for the best molecular weight for bioactive peptides. So we are on the right way. From our point of view, tomato seeds and brewery spent grains are the most promising feedstock for protein recovery. And in both cases, the enzymatic digestates contain, as expected, proteins and peptides, but they also contain sugars and phenols. So they are a mixture. Depending on the final putative exploitation, the fact that our products are a mixture can be useful. For example, if you are thinking to food ingredients, the presence of phenols can be positive or not useful. For example, if you are searching a specific peptides with specific sequence. The work is in progress. In case of tomato seeds, we are working on the tomato protein precipitation before the enzymatic treatments with the aim to recover more pure peptides without sugars and phenols. In case of brewery spent grain, we observed that peptides are already present also before the enzymatic treatment. This is probably due to the beer fermentation processes. So the yeast enzymes digested the proteins of barley before the feedstock production. So we are working on the recovery optimization of these peptides. Second example, of course, is Cipromed. In Cipromed, the work is in progress. Of course, we are working on 5 different types of feedstock, 2 varieties of Lupinus albus and 3 varieties of fava bean. In this case, we are adopting a different approach. We are working on protein solubilization from the solid feedstock with alkaline or neutral medium. So we will obtain protein dispersed in the liquid phase, and then we can separate solid and liquid phase, and we can precipitate by acid the proteins from the liquid phase to a new solid phase. These recovered legume proteins can be dried and used as they are, as whole protein ingredients, or these proteins can be further treated with enzymes, proteases enzymes, for bioactive peptide release. In both cases, they can be considered alternative proteins for food and feeds. Example number 3, it comes from a quite old project. It is BioRice is almost 10 years old. In that case, we worked with rice protein byproduct, and the aim was the production of bioactive peptides for exploitation in food, cosmetic, and nutraceutical sectors. Here you can see the rice starch processing flow schemes that starts from rice, of course, and comes to starch production. We worked on this particular byproduct of the industrial process that was a slurry, so it was almost liquid, and contains high amount of proteins, up to 17 grams per liter of proteins. This byproduct was treated with food-grade proteolytic enzymes to digest the rice proteins and to release peptides. These peptides were split into 4 fractions via cross-flow filtration according to their molecular weight. The best processes were scaled up and validated in bioreactor up to 10-liter volumes. Here you can see one example of gel electrophoresis analysis. Again, you can see that we obtained the low molecular weight peptide below the 10 kilodalton. And these peptides are mainly in the supernatant, in the liquid phase, so they can be more easily recovered. We assessed several bioactivity of this peptide fraction: antioxidant activity, antihypertensive activity, anti-tyrosinase activity. We also checked if these peptides were pro or anti-inflammatory, and we checked cytotoxicity and irritation because we want to avoid this activity. We also determined free amino acid component, and starting from these results, we evaluated the putative taste of this fraction because of food exploitation. And the most promising peptide fractions were further subfractionated, and we obtained the sequence of 365 peptides. Last example, Prolific project, another European project finished a couple of years ago. In Prolific project, we started with legumes, fungi, and coffee residues from agro-industrial, and we applied a cascade approach. It means that for each feedstock, we extracted first proteins and peptides, and the residues of the first extraction was extracted again to recover fibers such as chitosan and beta-glucans, and the residues was extracted for the third time to recover secondary metabolites. All these extracted fraction and the final residues were evaluated, were characterized, the bioactivity were evaluated, and at the end, several prototypes were prepared. Here you can see a summary of the processes optimized in this project from lab scale to pilot scale. In case of proteins, 3 major type of extraction were optimized: environmentally friendly aqueous extraction, enzyme assisted or ultrasound assisted extraction, and we obtained protein extract from chickpeas, peas, and coffee green beans. A wide range of tests were performed in order to rank and validate fraction functionalities according to the foreseen product prototype's final characteristics. At the end of the project, 16 prototypes were produced for food, feed, cosmetics, and packaging markets. Some example, here you can see 2 different examples of food prototypes. We have high protein bread that contains up to 20% in weight of proteins, and high protein pasta containing the chickpea proteins obtained in Prolific project. Again, other food products prototype example. In this case, they are vegan products. You can see high protein ester dates for surrogate foods to be used, for example, as snacks or for breakfast. And on the right, you can see a vegan burger. At the end, one example for another type of exploitation. In this case, it was a cosmetic prototypes. Active creams were produced, including phenols and peptides from coffee, and these creams had antioxidant and skin whitening properties. In conclusion, I explained you that plant proteins can be valuable alternative proteins for food and feed sectors, but also for other type of exploitation, for example, cosmetic or packaging sectors. Agro-industrial residues are a promising source of such proteins, with also reduced environmental and climate impacts. Environment-friendly and sustainable processes can be implemented, and enzymatic treatments are able to release peptides with improved bioactivity. Thank you for your attention. Let me thank also my group in Bologna University, including the students. Thank you.
Okay, thank you, Maura. Any question? Okay. So
Sorry, Annalise, I cannot hear you.
sorry, yes, because I have, of course, switched off the microphone. Okay, Christina, go ahead. I was saying that you are part of the Cipromed project.
Yeah.
Yep, so you can go ahead.
Thank you. Good morning. I'd like to show you an overview about one of the main risk factors when you talk about novel food safety, that is allergenic potential of alternative proteins. What we have to define first is the concept of cross-reactivity. It happens when a patient with a specific food allergy experiences allergic symptoms due to allergens derived from another food because of the similarity in both chemical structure and amino acid sequence between the two allergens. So in this case, foods are different, but usually they derive from phylogenetically related organisms. In the picture, we can see what happens at molecular level. On the left side, we have the schematization of the sensitization phase, during which allergenic patient immune system produce the IgE antibody against a specific epitope of the allergenic proteins. And these antibodies are able to bind the surface of the basophils or mast cells of the allergenic patient. So when the patient eats again the same food, the same allergenic food, the allergen can bind the IgE on the surface of the basophils or the mast cells, causing the degranulation of cells and the release of inflammation mediators such as cytokines, histamine, and so on. On the right part, we have what happens when the same patient eats another food with proteins quite similar to the primary allergen, a protein that shares some of these epitopes that can bind after digestion the same IgE on the surface of the mast cell or the basophils and can cause the same kind of degranulation and release of mediator of the inflammation, and so more or less the same kind of reaction. It is the allergic relation that could be local or systemic, it depends. So the idea is that if there are two allergens, two proteins derived from two different foods, but they can share some epitopes, these epitopes can bind the cell of the immune system of the patient, causing more or less the same kind of reaction. So, come to the project. In Cipromed, we are working on different novel food, edible insect, microalgae, and emerging legumes. Our strategy is well summarized by this picture that is from a very recent review. So we start from the novel food matrix. We perform protein extraction and characterization. Then we perform a Western blot with the serum of the potential allergenic patients. Then we identify by mass spectrometry the protein contained in the immune reactive bands, and then using bioinformatics tool and allergen database, we compare a structure and sequence of this protein, immunoreactive protein, with the already known allergens. Which are the target patient for our study in Cipromed? It depends from the food metrics. So if we talk about insect and microalgae, our target patients are house dust mite and crustaceans allergic patient, because insect, house dust mite, and crustaceans belong to the same phylum of arthropods, so they have some protein in common. If you talk about emerging legumes, for instance, lupin, the target patient are patient allergic to other kind of legumes, and then patient allergic to peanut, because peanut is the most relevant allergenic food for all the United States and also the north of Europe, so it's one of the most important allergenic food. And these patients are, these food are peculiar because it's a legume from botanical point of view, but it behaves as nuts from allergenic point of view. So it's very interesting to understand how what happens to these patients. Actually, most of the publication and also most of our work on this field is about insect till now, because there is already a position paper of EFSA that focused the attention on this aspect of cross-allergenicity about insect. There are many papers of insect primary allergy that came from research group of South America or Asia, because in these countries insects are common food. But there are also few papers about cross-reactive insect allergenicity, and all these papers focus the attention on tropomyosin and arginine kinase as cross-reactive allergens. Actually, there are already 16 tropomyosins and 7 arginine kinase from arthropod listed as food allergen or inhalant allergens. They have common chemical structure, a high sequence homology. The concept of sequence homology is very important in this case, because there are guidelines for cross-reactivity prediction that indicate that in a window of at least 80 amino acids, there must be more than 35% of sequence identity. So it's very strict condition. But what I want to show you is that sequence identity or structural homology alone is not enough to define a protein as a cross-allergen. You need to perform some in vivo or ex vivo test to verify that there is something more than sensitization. When I talk about sensitization, I mean the possibility that the IgE of the patient can bind a protein, an allergen. But you have to demonstrate that this binding can cause symptoms at clinical level. In this paper, for instance, they investigate 15 shrimp allergic patients, and they test them by skin prick test, that is the most common cutaneous test to perform allergy diagnosis. Then they perform basophil activation test. This is an ex vivo test in which you investigated the percentage of basophils that can degranulate when you put in contact with the extract, allergen extract. And then they perform also immunoblocking and double-blind placebo-controlled food challenge that is the gold standard for allergy diagnosis. And they found out that 14 patients are sensitized to this insect, to Tenebrio molitor, tropomyosins, and arginine kinase. That seems to be the more relevant cross-reactive allergen in insect, and 13 patients are positive also to oral food challenge. This means that it's not only sensitization, but they have symptoms when they eat insect. The problem of this paper is that there are few data, and most of them refer to a very small group of patients. Now I want to show you a paper from my research group. We tried to enlarge the patient population. We consider 28 patients allergic to house dust mite and 8 patients allergic to shrimps. And then we investigate 5 different insects: mealworm, buffalo worm, sealworm, cricket, and grasshopper, and 2 different kinds of processing. So we test the raw insect, but also the boiled and the fried. Well, what we found out is that most patients, IgE, immunorecognize at least 1 insect extract. This means that there is a species-specific recognition. So most of patients does not recognize more than 1 insect. Then we found out that tropomyosin is the most cross-reactive protein for both the patient classes, more than arginine kinase. And patient IgE recognition against tropomyosin is resistant to both processing. So this kind of immune recognition is thermostable. The last paper that I want to show you is quite different because it's about occupational allergy. In this case, we talk about primary insect allergy, is again by my research group. In this case, we have 10 people employed in a breeding of Tenebrio molitor and Hermesia illucens. 2 of them show symptoms against Tenebrio molitor larvae, but also Tenebrio molitor faces. In this case, the route of sensitization is not by ingestion, but by contact or inhalation. We perform skin prick test against house dust mite and different type of crustaceans in order to understand if there is some cross-reactivity, but both the patient result negative, so it's not this kind of reaction. And then when patients that travel a lot in the East country try to eat insect, they have oral allergic symptoms just against Tenebrio molitor and immediately after the first ingestion. So this means that there is some sensitization process before. We perform basophil activation tests against Tenebrio molitor larvae and faces, and both the patient result positives. And then we perform immunoblocking to understand which is the allergen or the allergens involved. And we found out cockroach allergen-like protein. That is completely another kind of allergen, different from arginine kinase and tropomyosin, because it's not a case of cross-reactivity where only the protein that share sequence similarity between organisms are involved. In this case, it's a primary insect allergy that is, again, species-specific because the 2 patients have not any problem when eat other kind of insect, but also the allergen involved are different. So in conclusion, patients allergic to house dust mite and crustaceans should be careful when eating insect. They can have problem with 1 insect, but not with another one, but they can be careful. Primary insect allergy and also cross-reactivity are species-specific, and the insect allergen for which there is solid evidence of cross-reactivity are tropomyosin and arginine kinase. Maybe there is other one, but these are for sure involved. Thank you very much.
Okay, thank you. So, are there any questions for Cristina? Okay, there is a question in the chat.
Yeah.
I would like to ask you if it is possible that the level intensity of allergy in insect may alternate based on the form of the final product derived from insect, dried, whole insect, insect meal, grinded, dried, insect protein, insect oil, etc. Okay,
Yes,
so
we
you can read it in-
investigate. Yeah, we investigate processing for insect, but also for other food metrics in the past. And it depends. I mean, there are allergens that are very heat stables and also stable at digestion and everything. So they affect also if you process the food a lot, but there are allergens that are heat labels. So you can, if you cook, if you perform processing, they lost their allergenicity. So it depends from the patient, because there are patients that are allergic to a singular allergen, and so the difference is if it's stable or not at processing. But there are also patients that are sensitized to different allergens and also different foods. So it's more complicated in this case.
Thank you. Okay, so there is no, I was saying, there is no way of predicting a processing method to delete or reduce the allergenicity, because you don't know who is going to eat the food.
I think that it's not possible to use processing to eliminate allergenicity. So to go to the market with not allergenic food, but you can use processing, and we clinicians do this to improve, to use a hypoallergenic food to improve the process of induction of tolerance to desensitization to an allergenic food. So this is a process the clinician use for children when they try to induce the tolerance to a specific food, and starting from a high processed food that normally is hypoallergenic is a good way to start.
Okay. Okay, so thank you very much, Christina. Of course, also for Christina, if you have further questions, just write them or mail me or mail Christina directly. You find her. Okay, so we are at the end of our webinar. It was, I may say, very, very interesting, very diverse and very interesting. So again, if anybody needs a paper saying that they did participate to this webinar, please mail me and I will send you the proof of participation. All the material will be in some way made available. We have recorded the presentation, so I ask to everybody if you have something in contrary to share your presentations, where we might not share it completely, but we will, well, okay, anyhow, we will let you know what we are going to do it. Thank you so much. I don't know if Shanes, do you want to say something in conclusion, or Christina, or I would say thank you for everybody that did participate, or Christos also, I see you.
Just thanks as well. Well done.
Yeah,
Yes.
thank you. And I find it very interesting to have this common webinar between the European projects. I think it's something that we need to repeat.
Yes, yes, we can do something more. Christos,
thank
you so much
for the coordination of this really interesting
No
webinar.
problem. It was easy. It was easy. Thanks so much to everybody. And
Thank you.
so we will do some more webinars, okay, on both projects. So the web pages of both projects are there to give you all the updates. So thank you so much and see you soon. Bye.
Thank you. Bye.
Thank you. Bye.
Bye bye.
Bye.